Thank you for this introduction. And I would like to thank Dr. Ammar Al-Banna for giving me this opportunity and arranging this amazing conference. It's really a big learning opportunity for me, getting exposed to these nice talks from giants of neuropsychiatric, clinical aspect of it, at least, which I wasn't familiar with in-depth knowledge. So what I will talk about today is basically the genetics of autism spectrum disorder, the current status of it, what we know so far, and what we can infer in terms of etiology of autism spectrum disorder, and what are the roadmaps, the future roadmaps where we can go from here. So first, I'll go through the heterogeneity part that we observe clinically. And this heterogeneity of autism spectrum disorder, you can subgroup them in two major or broad categories. One is the structural and functional aspect of these comorbid conditions that we often see manifest with autism spectrum disorder, and the other subgroup is the functional. So within the structural and functional classification, microcephaly, epilepsy, heterotopia, or focal cortical dysplasia, often comorbid condition, often manifest as a comorbid condition with autism spectrum disorder, and these conditions have sometimes have physical abnormalities or at the tissue level in the brain. Whereas ADHD, OCD, or non-syndromic intellectual disability or learning disabilities doesn't have any tissue-level dysmorphic feature in the brain, but they are functional at the level of synapses. You see deficits. So there is a wide range of heterogeneity exists in autism spectrum disorders. And we know it's a multifactorial condition, and genetics is a big part of it, so is environmental influences. And along with Dr. Bennett, I also agree there is no evidence whatsoever MMR vaccine has anything to do with autism. But the environment and genetics, they interact each other. We don't know how and at what level, but the interaction you can assess through statistical inferences. So my talk is all about genetics. So how much of it is genetic? So from multiple studies, multiple twin studies, I would say, the contribution of genetic portion of autism ranges from 70 to 90%. So 70 is the lowest and 90 is the highest that has been reported. And this diagram was drawn from 70 to 70, considering the 70%. And 10 to 30% is non-genetic cause of autism that we know nothing about. So the rest of the talk is about this genetic portion of it and how much information we can infer regarding autism etiology. So I'll start with what do we know about genetic mutation. So we'll start with cell. Inside the cell, there are chromosomes, and within the chromosome, DNA wraps around, and we can have mutation in the DNA, and mutation comes in different forms. So genetic mutation can happen at the level of chromosome. You can have an total extra chromosome, and also in the reference genome or reference DNA. These are two genes, and in someone's DNA, one gene can be deleted, or someone else's DNA. The second gene can have a duplication. So these are called copy number variation. It changes the number of copy of the genes in our DNA, and when the copy changes, we think that the level of protein also changes because the number of functional unit either it decreased or increased due to this event. Now, the other form of mutation is called single nucleotide variant. It's at the point level mutation. You see a change at one base. And there's another level of mutation that happens like 5 or 6 base pair long. Those we call small insertion and deletions. So these mutations can be, been in terms of their length. So this is a karyotype of a Down syndrome patient where you can see the trisomy 21 chromosome has been duplicated. And this can be targeted by karyotyping, and the copy number variation usually in terms of length, it starts from 1 kb around that range, and maybe 10 kb, 10 megabase in length, because beyond that you can do it by karyotyping. And for single nucleotide variants and insertion and deletion, you need a more sophisticated technology. So for the first two, we can do clinical microarray to identify the large structural differences in our genome. But the idea was to develop one test to profile all the mutation in our genome. So clinical microarray was not the one that we should go for. So now what we have, we call is whole genome sequencing. So whole genome sequencing is a technology that sequences the entire genome of our that we carry in our body, and it will profile all the mutations. Even though we are not there yet with copy number variation calling, but most of the single nucleotide variants now can be profiled with a very short time. I'll give you an example. The first genome took 13 years to sequence, and billions of dollars spent in USA just to make the blueprint. Now you can sequence multiple genomes in one day. So the mutation that we see can be inherited from parents, or it can be de novo, meaning new mutation. So the most relevant mutation for autism spectrum disorder is actually the de novo mutation. So what happens is this mutation you will not find in parents' blood or any other tissue. They arise in the germinal cell, oocyte or spermatocyte have this mutation, and these, after mating, the zygote acquire this mutation from the germinal cell of the parents, and this mutation then remains for rest of the body in the offspring. So you can find this mutation in the kid's blood or any other tissue, but you will not find the same mutation in parents' blood or any other tissue. So this mutation can be chromosomal level, single nucleotide variants level, or copy number variation. So what does it mean in terms of autism? So this is just a depiction, a cartoon diagram. It's not quantified in any way, just to let you know. So we have our background, genetic background that we carry with us, and that may expose as a risk factor to some level. But then there is a big, if you think the risk factor is a cup, and there is a threshold for risk factor, this de novo mutation, actually the rare mutation with high impact of risk factor, and when somebody carry this high impact de novo mutation, it crosses the risk factor threshold and manifests the disease. So in autism spectrum disorder, de novo mutation carries a large volume of the risk factor. So this is what I'm going to talk about today, the contribution of de novo mutation in autism spectrum disorders. So genetic test, there is a consensus about genetic test in the clinic. It's considered to be the first-year diagnostic test for developmental disabilities and congenital abnormalities, and this consensus was published in 2010, and it's every major hospitals in North America, Europe, and in this region these days we are doing a regular genetic test to find out these variants, specifically large variants. So what we know about the diagnostic yield, I'll just move with the diagnostic yield first, then I'll show you the types of mutation and what we know so far in terms of etiology. So what is the diagnostic yield? If you apply whole genome sequencing, microarray, or exome sequencing to find out pathogenic mutation from kids impacted by autism spectrum disorder. So the current breakdown of diagnosis is this. Single nucleotide variants have been reported to contribute around 18 to 30%. in different publications. So this diagram is the conservative diagram, considering the lower threshold. Karyotype contributes 5%, and also rare copy number variance contributes 15%, even though a recent paper came out showing that they can identify 24% of kids with autism and neurodevelopmental disorder having a pathogenic variance. So if you if you are very conservative about this, still it is 38% of the kids we can identify a pathogenic variance, and that I think is a big step forward. Because the whole genome sequencing technology has been introduced only in 2008, 8 years ago. Now we can detect 38% of the variance. That's a very big turnaround. So now the mutation, what do we know about this from this 38%? So about the etiology of autism spectrum disorder. So I'll show you my experiences from different projects and what we know. So this is the biggest project I was involved in. Is called Missing Project. So we have conducted whole genome sequencing in 5,000. So that's the number that we published, but the actual project aimed to do 10,000. We are writing the paper now, it's been sequenced, but this data is from 5,000 samples. So from the 5,000 samples, these 2,000 of them are male and 559 are female. So it's kind of comparable with the 4:1 ratio that we know, the bias that exists. And we also have a good number of unaffected siblings that we have sequenced. So we cannot do it alone. It's too big. So Google chipped in, and they are holding the data for us. It's multi-petabyte level data. Even sick kids cannot handle. Sick kids, we are the work engine for it to analyze the data, and Autism Speak, sick kids, and Google funded the project. All three of them. So it's a multi-million-dollar project. Came to an end, and I'll show you what are the results. So from a whole genome, how many de novo mutation we can have? It's 73.8 to be exact. That's the de novo single point mutation we have. And from this 73, one exonic de novo. So anybody, not just kids with autism, any normal people here in this audience have a one exonic de novo mutation you carry that your parents do not. And we also have a 12.6 de novo insertion and deletions and large copy number variation changes in our genome. So what does it mean when in terms of autism, these mutations? So when we lined up the parental age and the number of de novo that we detect from the kids, that's a very interesting finding we have. The parental age for the father, the paternal age, is super significant with the number of de novo mutation we observe. So in layman's term, this figure shows that the more age the father is, the kids will have more mutation, a de novo mutation. Part of the reason is because germinal cell, especially sperm, goes through multiple toxic environments, and the spermatocyte, the cell division, when it happened, may have introduced to some environmental stimuli that acquire de novo mutation. So with age, so that's why the kids whose father is age enough will have more mutation. But that's just an association. It's not true all the time. So we see an enrichment here. But this enrichment has been replicated in 4 or 5 other labs. So it's very solid data right now. So how to interpret the mutation that we see in unaffected siblings and in autism kids? So what we did, this is a gene, and these are the functional unit of the gene called exon. So what we did for each exon, we computed the level of expression for this gene. Expression means the functional activity that we see at the mRNA level. So we are not looking at gene level, we are looking at really narrowing down to exonic level activity of the gene in the brain. And we also look at how much population mutation that we have in that particular exon. So how much activity going on in the brain and how much population burden. Interestingly enough, there is a negative correlation we saw. So the more burden that we have, the less active in the brain. So it's never been shown before. So some of the exon in the gene is a very interesting property. So we patented this. And this shows that some of the exons are really highly active in the brain, but in normal population, they don't acquire any mutation. So that's very fascinating. But what we did, we overlay the mutation that we see in unaffected siblings and autism, kids with autism spectrum disorder, and look what we found. We found a huge enrichment of those de novo mutation impacting those exons that we see highly active in the brain, but not accumulating mutation in the control population. So we call them critical exon for brain. So the critical exons are hammered by de novo mutation in kids with autism spectrum disorder. So that's a very interesting observation that we have. And from this, what we did, we gathered data from 536 postmortem brain samples from 16 different brain regions to find out these critical exons within numerous genes who, that are impacted by de novo mutation in autism, which brain region they are highly active. So this diagram shows the de novo mutation show enrichment of critical exon that are highly active in prefrontal cortex during prenatal development, because our brain data also came from prenatal brain. So those are donated by the parents. So this shows a large number of genes that are impacted by de novo mutation are prenatally active. So it kind of supports the hypothesis that the autism spectrum disorder manifests very early in the gestation stage. And what we did, we identified all the genes that have critical exons and have a mutation, and we want to see what pathway they're involved in the brain. And this is just a pathway diagram from many, many analyses. We show that these critical exons are at the core of the neurogenesis. It involves in neural synapses. It involves in voltage-gated channels that it requires for communication within the synapses, and also neural differentiation. So there are multiple clusters that are significant with the red dots. These are the clusters that are involved in neurogenesis. So this finding has been, after finding this, we patented this as a method to, classify the mutation. And some of the companies actually bought this, and they're using it regularly for their report. Not to say this mutation is causal, but they can now point out the mutation. This mutation is active in what brain region and whatnot. So I'll jump into the next study that we have done from this big project. And one of the questions we asked is there any correlation between autism spectrum disorder morphological severity and mutation? So the behavioral severity is hard to judge, but the morphological severity is really easy because it's physical dysmorphic feature that you count. So what we did from the whole data, we kind of carved out 258 constitutively ascertained unrelated children within Canada. It's from Newfoundland, where I did my PhD and all my education. And this is a homogeneous population. It's an excellent population to do genetics. We have screened whole genome mutation profile for all these kids. And also, there is a published paper where they can quantify major congenital abnormalities and minor physical anomalies. So we kind of apply that quantifiable approach. Basically, it means the more score a kid have, that means the more morphological sign he carry. So we want to correlate now. Is there any evidence? I think there was a question like, can we subgroup the kids so we can have a genetic profiling? So this can aid taking that decision, even though it's one single study, may need to be replicated. Actually, the replication is going on right now. So what we find is de novo mutation with low severity, low morphological severity is low. But the high severity in morphology have high number of pathogenic de novo mutation. So there's the y-axis is the number of de novo mutation. protein coding mutation. So this kind of shows that if you have morphological cases, maybe sequencing is a very good idea, or doing genetic tests may be a very good idea. So this has been published in JAMA. So then the other question is, from this typical type of mutation, can we guess something very new in terms of mutation and the predisposition of those mutations into autism? So this is kind of like a interesting paper came out in New England Journal, showed that there are patches of disorganized tissue in the neocortex in children with autism. So they didn't elaborate much, but they showed with staining with immunohistochemistry that the cell didn't migrate properly. So there are patches of the tissue that looks different than the normal part of the brain. So we have this is just an example. We have this complex case where the boy is 6-year-old male diagnosed for focal onset epilepsy with autism spectrum disorder at sick kids. And the MRI imaging shows the flare signals. And so the kid was having really bad time with the seizure. So he used to wear a helmet. So what the neurosurgeon did, they took out the skull and put the grid on him just to find the focal point of the seizure. So after a few days in the hospital, they found the focal point for the seizure, and they intervened, and they took out the tissue. That's the tissue that we got in our lab for analysis. Then we did the sequencing, and in the blood and the overall tissue, we found a deletion in STXBP1 gene. So STXBP1 gene is a bona fide gene for epilepsy. It's a known gene it's the only gene that has a 1% threshold for epilepsy, actually. So we are very happy to see the deletion, but things got interesting when we did the profiling of the tissue. When we dissected the tissue, we did staining with H&E staining and also neon staining. You can see the cluster of neurons, small dots are really close together. Those never migrate. So the normal neurons should be spaced out like this, but you can see with multiple staining, there's many neurons from that resected brain tissue never migrated properly. And okay, so we did sequencing on those tissue, and we found that those tissue doesn't have that heterozygous mutation. They have a homozygous deletion. So it's a totally different profile we have identified for those tissue where the neuron never migrated properly. So this kind of shows that cancer-like mutation, somatic mosaic mutation, can predispose to neurodevelopmental disorder. So we went back and reanalyzed our whole genome data, and interestingly, what we have found is 5% of the autism kids have a somatic pathogenic mutation in their DNA. And those are high-level mosaic mutation because at least for 15 to 35% of the brain cell have been impacted by this. And I'm writing the paper. No, it's not published yet. These high-level somatic mutations arose early enough that they must be present in the brain. So this is an example. This is one of my favorite example that shows that the kids, the two siblings, have a germline deletion and PRP1 de novo stop gain. But the dad also carry those deletion. We thought that is de novo, but it's not de novo. And also PRP1 stop gain that we see in this kid also found in 20% blood cell in the dad. It's not into it. There's a renin damaging de novo missense mutation in 60% of the blood cells. So what it shows, we carry different genetic profile in our own body, not just one genome. We have multiple genome in our body. So the future is we have pathogenic mutation detection is going on very well in terms of sequencing and stuff, but the therapeutics is really awful. We do not have anything. So that's the future. So understanding the pathobiology of these mutations. So this is one example where 14 Q13.3 microdeletion syndrome, where we did a knockout mouse, and we see this different level of arborization in the brain, and it has been replicated in vivo and in vitro. I'll just move forward. This will be my last two slides. So there's two recurrent mutations we found in green 2D gene, and these are pathogenic mutation, but this gene is involved in NMDR channel regulated by synaptic transmission through the use of green 2D protein. Interestingly enough, FDA approved NMDR antagonist. So they applied, after detecting this mutation, they applied that approved drug, and they saw some improvement. So that's the last slide I have. So this is the future. We think that we can have an isogenic model of human cell lines, and we can apply the mutation into the isogenic cell line through CRISPR machinery, and we can differentiate them into primary brain cell, the mutated line, also the control line. Then we can characterize to identify the right target for therapeutic intervention. So I would like to thank all my supervisors and centers that helped me coming this far with doing these researches. Thank you.